This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1127, recorded on June 28th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and hello, everybody else who's on this show. And that happens to be another person, but I don't want to mention their name because I'll step on his lines. It's a beautiful day. It's an unbelievably beautiful day out there today. It is almost zero humidity. It's actually a little bit chilly. Just consider last week, for instance. I monitor the temperature in my local trout streams, and one of them, for the first time in my memory, hit 90 degrees. A trout stream that's 90 degrees. That doesn't work.
Where, at the surface? At the surface?
No, it's hard to measure it. But no, you measure it at the bottom of the river.
So the trouts are not very happy. It all
mixes together anyway. It's all pretty homogeneous in terms of
the trouts are not happy at that temperature, are they?
Unhappy is the least of... I mean, they might die. You could get a fish kill at that temperature for trout. And for some lakes, too, because there's not a lot of oxygen you can dissolve in the water at 90 degrees, right? And that's the biggest problem. So, at any rate, other than that, there's just such a contrast compared to that. Quite wonderful.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is also gorgeous here, and we are very, very happy about that. By the way, a trout stream at 90 degrees Fahrenheit, I believe, is referred to as a bouillabaisse. Yeah, so we have 74 Fahrenheit, 23 C, a few puffy clouds. It's gorgeous. It's supposed to be pretty nice tomorrow, and then start pouring rain again.
All right. Dixon, I think you were just muted. Yeah, you're muted.
There you go.
Are you okay, Dixon?
I'm fine. I would like to go and close my door because some people just arrived, and I would rather not have that as background noise. I'll be right back.
Go ahead. Yes.
After this brief message from our sponsors.
Well, if you enjoy these programs, including Dixon getting up to close the door, consider supporting us. Microbe TV is a 501(c) nonprofit entity, which means in the U.S. your contributions are federal U.S. tax deductible. And we depend on your contributions to do our work. We have a nice staff now that helps out with all aspects of recording and production. We have a studio in New York City. We'd love your support. Go to microbe.tv/contribute. All right, we have a couple of news items today, which make my blood boil like the waters of the trout stream. Oh my gosh. Number one, in Reuters, Pentagon ran secret anti-vax campaign to undermine China during the pandemic. Oh my gosh. Apparently, the U.S. military launched a program to discredit China's vaccine, which is Sinovac. And this was apparently in retaliation for Washington calling it the China virus, right? Yeah. Which our president at the time did, right?
Well, no, wait, what was it? What was in retaliation?
We tried to discredit.
Yeah, we tried to discredit Sinovax.
Right. And the reason was that it was because the U.S. had blamed, that's not the reason. That doesn't make any sense. Why did we do this? We launched a secret, to counter China's growing influence in the Philippines.
This is not, so just to be clear, the Pentagon in general, I mean, sure, at an individual level, they're humans, but as overall strategy goes, they don't like try to play tit for tat type of games. This is a strategic move that somebody decided China is gaining too much influence in the South Pacific, and we don't want the distribution of their COVID vaccine to influence, say, people in the Philippines to favor
Right.
China because, China provided this vaccine. And so apparently the U.S. military had this secret campaign to sow concerns and to discredit the Sinovac vaccine so that people in the Philippines would think that it was bad.
Right. So they flooded social media with all kinds of messages, including like this one. COVID came from China and the vaccine also came from China. Don't trust China. That's where Mike got this idea about China virus. But that was a typical tweet.
Is Sinovirus vaccine also an RNA-based?
No, it's an inactivated.
Sinovac is an inactivated vaccine.
I see. Effective?
No,
not particularly.
It's not.
So why did we bother?
Certainly, right. So certainly
we
when you
didn't
have other vaccines on the market that are 90% plus effective, Sinovac didn't look all that great. I was actually rooting for Sinovac. I love the simplicity of using an inactivated vaccine, and then the results came out. It's not terrible, but it's not great.
This campaign began in the early 2020 before we knew anything about the efficacy of the vaccine. Anyway, it started under Trump and it was continued under Biden, who eventually stopped it.
Well, okay, so it stopped in the spring of 2021. Biden was not in charge until the end of January 2021. So let's not. I'm not entirely clear that he was on board with this.
Yeah.
It says it continued months into Joe Biden's presidency.
It did, but taking over as president is a rather complex job, and I'm not sure at what point he found out about this.
Yeah.
Anyway, it did. Yeah.
Spring 2021, he said he banned it.
Yeah.
Anyway, obviously this is not great, right?
No.
I mean, vaccines, everyone needs vaccines.
You got it.
And it's not good to sow this kind of misinformation.
Right. As an aside, I always watch the evening news and PBS, and night before last, Paul Offit was on.
Right.
And he gave a great summary of why anti-vaxxers are anti-vaxxers and why vaccines are essential for human life. And presented a black and white view. You can't take sides in the middle on this one. You have to be one or the other. But if you take the other, you're sort of anti-human. Yeah, he was excellent, though. I mean, he actually wore a suit, which I thought was unusual for him.
Anyway,
adapt to your audience.
There's a quote here from Daniel Lucy from Dartmouth. I'm extremely dismayed, disappointed, and disillusioned to hear that the U.S. government would do that. Completely indefensible. I would agree with that.
Well, did a lot of other bad things. I don't want to even go there.
Well, sure. I understand what the thinking was behind this, but I wish somebody higher up had looked at it and said, no. Yeah, we're concerned about China's growing influence in the South Pacific, but we can't address it this way. This is just not.
Do you think Tony Fauci knew about this?
Oh, gosh, no. No, he was not in on secret Pentagon briefings. No.
By the way, his interview on the public television was in 3 parts about his new book that just came out, and he was remarkably candid and restrained. I would
He's
say.
Allowed to be now.
Yeah, I know that.
He's off the chain.
How was he?
Gag limit.
How was he, Dixon? How was he candid? Give us an example.
He didn't actually speak out negatively about Trump. What he said was that he ran into some. He said they started out great friends, and then as the relationship evolved and Trump realized what Fauci was in control of, the relationship broke down. And he said there were major philosophical differences between myself and the president, and he would not go further. He would not call him a liar or rat or a rapscallion or any of those other terms that he's deserving of but didn't get. I think Tony Fauci is a gentleman, but a tough Brooklyn-raised gentleman. And remember, he said, "Don't let the bastards knock you down." He said that's what someone told him once when he was a kid. He never forgot it.
Okay. All right. Our next story from the Associated Press. New York governor's subway mask ban proposal sparks debate over right to anonymous protest. So, a new proposal by Governor Hochul, I don't know how to say it. Hochul, to ban masks on the New York City subway. Now, the reason she was doing this, in fact, before COVID, there was a ban on masks so that people can't hide their identity.
Yes.
And in the current climate, they're worried that people come on subway trains, put these masks on and yell at people. But for gosh sakes, infectious diseases. If you want to protect yourself, why can't you wear a surgical mask or an N95? I don't think this is right. And in some ways, a place you'd want to wear a mask because a lot of people on it, right?
Yeah, that's true.
Yeah, I think this is poorly considered.
I don't think it's enforceable.
Oh, it could be. I mean, they've got cameras all over the subways. They can see people. And if you're wearing a mask, they can see you're wearing a mask. So,
New York previously banned public mask wearing by groups of 3 people or more under a law dating back to the 19th century, when upstate tenant farmers dressed as Native Americans and rose up violently against landlord. That law was lifted when COVID-19 began. So if I'm the only one on the car and I wear a mask, I'm good because it's not 3 or more, right?
Yeah,
I get on a subway car and somebody over there is hacking away. I'll put a mask on, right?
Yeah, I think if it were implemented in something like that, where it would have to be groups, I mean, this still wouldn't address the issue of an individual coming onto the train in a mask and doing something. But there are other laws that would outlaw, things that people would do.
Yeah.
And if you're trying to stop somebody coming on and shouting anti-Semitic remarks, I don't think you can really do that because somebody can say anything they want.
Sure.
As long as they're not anything they want. They can't incite a riot. They can't yell fire when there isn't one. I mean, there are limitations on the First Amendment, but I think this is poorly thought out.
I would agree.
And this is legislation that hasn't even been written yet.
That's true.
So I think
if you go to Chinatown, for instance, in New York City, half of those people are wearing masks.
Sure.
It's almost tradition to wear a mask. And so, how do you legislate against that? I mean, I don't think you can. Really,
no.
Well, if they do, I'm wearing a mask when I want to.
Good.
So you guys might have to bail me out.
Okay.
Remember, 3 free meals a day. Don't think about it.
All right.
Get bailed out.
Our next article. Colorado bat facility that will support virus studies sparks outbreak fears. This is like a general thing happening in the world now. This is a University of Colorado State University, sorry. It's a facility where it has a bat vivarium where they're hosting a number of bat species. And it's going to be a resource. They're going to provide bats and bat tissues are going to be bred there, right? So they're all going to be checked for pathogens. They're going to be bred there. And then they will be used for studies there. And also other people need tissues and so forth. So obviously, this is going to be important because bats harbor a lot of viruses with pandemic potential. But people are objecting to this. There's this organization called the White Coat Waste. It's absolutely reckless to bring non-native species of bats who carry deadly viruses onto U.S. soil where they don't exist and manipulate them in dangerous experiments. Okay, they're brought onto U.S. soil, but they're checked for pathogens. They're not manipulated. Oh my gosh, this is completely with having an agenda, yet not thinking through what actually is going on.
Well, they should object to the labs at Fort Collins also, then. That's an arbovirus laboratory for viruses that don't exist in this country. And they experiment on them lots of times, like Japanese bee encephalitis and those things. So you can't really find
Yeah.
logic to any
There are
of
many
that.
labs. Not many. There are other labs in the U.S. with bat colonies, like Rocky Mountain Laboratories.
Listen, the Bronx Zoo has bats.
Dixon, there
It's
are
in their
bats in your
world of
attic.
darkness.
It's also worth considering the source. This group, White Coat Waste, is a sort of PETA adjacent group. They're against all animal research, and they're trying to ban animal research.
As a woman who lives near the vivarium in Colorado said, the construction was pushed through without any public input. If lab workers have got gloves that have been cut open or people have been exposed, these people shop at the same grocery store that I do.
Alan, come on, a blast from the past. What song was this line from? Paranoia will destroy you.
Oh, that's that's the Who?
I don't know, but it's a good line.
I'm going to look it up. Paranoia will
destroy you.
The Assembly.
Really?
Oh.
Or the Kinks.
The Kinks. Oh, okay.
The Kinks. Aha.
Yeah. Anyway,
yeah. So this.
Go ahead.
This is a facility that's going to be doing federally funded research and is therefore subject to all of the federal protections on animals. And, somebody gets a rip in a glove, that is an incident that they would have to report. And there's a form and there's a process and this is overblown. But it is there. People are getting whipped up into a frenzy about it. I don't know how many.
Of course, White Coat Waste has gotten records showing that, there are biosafety incidents occurring, but none of them amount to anything because they're controlled. The university reports everything, and in all cases, there is appropriate reaction to the incident. So this is just fake reasons not to do this.
There are aviation incidents occurring all the time. If you go, NASA maintains a database, the Aviation Safety Reporting System, and it has, it's just, you can find tons and tons and tons of reports of things that went wrong on flights, and it's still the safest method of travel ever devised.
We have astronauts. We have astronauts waiting to come home.
Right. In fact, in large part because of all of these reports, that people are able to address.
I was on a flight to Europe a couple of weeks ago. The pilot said, "I'm sorry we're delayed, folks. So one of the seats broke, and we have to fill out a lot of paperwork."
Yep.
And plus, they have to rope off the seat so nobody tries to sit in it, right? Wow. So we were 30 minutes late because of that.
On my flight home from ASV, the second leg of the flight, I was sitting at a seat that had a piece of tape holding the tray table in place, so it couldn't be. And the tape had a set of numbers on it that I recognized were technical service order (TSO) numbers, indicating this had to have been FAA approved tape that they used to secure the tray table that broke. One tray table on an airliner, and I'm sure somebody had to fill out a form for that. So there are incidents occurring in animal facilities.
Happens.
And yes, you can find those reports.
Now, I have actually visited this facility at Colorado State. They have already a bat colony, which I got wrong because Tony Schounts, who is there, wrote to Twitter, said they're not that bad that you said, there's something else. But anyway, it's extremely secure and you can't just do anything you want, and it's all useful experiments. So just chill, folks. Let people do experiments. All right. And there's more. There's more. One more. House budget bills offer little good news for U.S. science agencies. So this is an article in Science. So basically, it's about the House releasing a preliminary spending bill for 2025, and included in that, of course, is science funding. The NIH, surprise to surprise, its budget will not go up. It's going to stay at $48 billion. But the bill includes a ban of funding of gain-of-function research, an outright ban of gain-of-function research. Now, I don't know what they're thinking. They're probably thinking that there's only one kind of gain-of-function research, right? But just about every lab, every biomedical lab, including those that did the papers that we're going to talk about today, do gain-of-function research. It's giving an organism a new function.
How about fixing somebody's broken leg?
I don't know if that's a gain-of-function, but if you put a piece of metal in, it's probably a gain-of-function, right?
Absolutely. Absolutely.
I mean, just about everything. Every organism is subject. So this reflects to me, and we have a letter about this that criticized our position last week,
Right.
saying, lawmakers shouldn't participate in science regulation. Well, this is a reason why they shouldn't, because they didn't even know what gain-of-function is.
Yeah, okay.
They want to ban it all. So what would be the effect if we banned all gain-of-function research in the U.S.?
First of all, this is a draft budget.
Okay, I'm getting excited for nothing, but
there's a whole process that goes on here. Secondly, there is a lot of political pressure to do something about experiments that are considered risky. And so somebody decided to take a stand. If this goes through with the ban on gain-of-function research, then it will be up to congressional staff, but also agencies to figure out exactly what that means. So there's a lot of wiggle room on the definition here. I'm sure what comes out at the end is not going to be a stoppage of all research.
Mm-hmm.
The flat budgets, I mean, that's a function of the federal budget. This happens. The NIH and NSF and all the science agencies are subject to this variance of budgets from year to year. All government funding happens that way, and it's lean times, so they're restricting the budget. I wouldn't make much of that. The thing with restructuring the NIH is that I think is not something that should be done at the congressional level. That I would agree with you. I mean, a lot of people could have input on that process, and in fact, many people who definitely know what they're talking about have commented that the NIH has too many institutes, and that might be a good idea to try to streamline some things.
Yeah,
but I don't think
fine.
a fiat from the House of Representatives is the way to go with that.
Yeah, I think streamlining is fine. We actually talked about it last week, but even in that bill, there were talk about regulating gain-of-function research. They wanted Congress to play a role in giving out funds to laboratories. I mean, that's not the way it works. Congress gives NIH a pile of money, and then they give it out as they see fit. The
whole point of having study sections is to decide how the money gets distributed.
Yeah, exactly.
Yes. But I don't agree that non-scientists should have no input on the topic of gain-of-function research. It's something that people feel strongly about. The people who are paying for it feel strongly about it. And that's something that, we as scientists need to explain and present the case for. And I think here on TWiV, we've tried to do that, but our audience is not the entire voting public of the United States, sadly.
I think non-scientists could have a voice as long as they understand what it is.
Right.
We'll give you a test.
No.
Tell us what gain-of-function research is.
A major issue with it is to define what that means. And I think once you get people into that conversation and start talking about what is useful and what is typically done, they can come around to the understanding that, there might be research that needs to be watched closely, but you can't just ban a whole category with a vague term like that.
Right.
I also think that much of this dialogue is based on this incorrect idea that the virus came from a lab. And now the lawmakers are saying, this is really bad, so we have to ban it. And this is just a bad starting point, right?
Yeah.
I don't know what to do about that. But I mean, yes, education is the way to go. I agree we should have dialogue, but many of these committee hearings, they're not a dialogue. It's accusatory, right? Could you, Dr. So-and-so, can you explain what gain of function research is? Just a simple question like that.
Yeah.
That would help. Okay. I know it's just an idea, but it bothers me.
Yeah.
And I'm sure it bothers all of us because science is key to our lives, right? We don't want to mess it up. And I've seen over my career, I started my career, there were a few regulations. And with each year, there were more and more. And now, in the last 10 years, this idea that scientists are unsafe and need to be regulated, I don't know where.
I remember when I was a graduate student at Notre Dame, the federal government came around with a variety of decals, and they asked us to choose the one we thought best represented biohazards. And of course, I didn't pick the one that they they had some bizarre shapes, but that's the way it was done. If that was how recent that actually started, that's 1965 or 66, where they were starting to say, well, maybe we should put a notice up on the board that there's something dangerous beyond this door in that incubator, and you shouldn't go in. So yeah. Today, we've got books filled with, you serve on committees, right? That actually, the biosafety hazard committees, and oh my God, it's a lot of red tape now.
But we are also doing a lot more research now.
And we are, and that's right.
And we know more about the risks involved.
That's right. And plus, there are some classic examples of laboratory escapees, which we'd rather not talk about, but we have to.
Yeah.
So there you go.
All right, now we will talk about the science that is done, funded by places like the NIH. Right. Very interesting science. We have 2 papers for you. The first is Nature Communications, Evolution of STAT2 Resistance to Flavivirus NS5 occurred multiple times despite genetic constraints. And this comes from Veit, Salim, Jung, Richardson, Boys, Quinlan, Baral, Bednarski, Hamilton, Kikawa, Nels Elde, Adolfo Garcia Sastre, and Matt Evans. And they are at Mount Sinai, the New York City, University of Utah, University of Washington. I think that's it. And of course, Nels is on TWiV here at Microbe TV. Okay. Now, viruses infect cells. The cells make interferon in response very quickly, and interferon is a great barrier to infection. So viruses have proteins that antagonize interferon responses. If they didn't, they wouldn't exist. That's how good interferon responses are. And the flaviviruses, the vector-borne flaviviruses, like Zika virus and dengue virus and others, they encode a protein called NS5, non-structural protein number 5. It also happens to be the RNA polymerase of the virus and a methyltransferase and an interferon antagonist. And what the NS5 protein does, very interesting. It binds a cell protein called STAT2. STAT stands for signal transducer and activator of transcription. This is important when the interferon binds receptors. The eventual effect is that the cell makes a lot of antiviral protein called interferon-stimulated genes, and STAT2 is a key part of that process. And so NS5 messes with the function of STAT2, so the interferon-stimulated genes can't be turned on.
Right. So one of NS5's functions is to stop the signal that would otherwise turn on interferon.
And many flaviviruses, they're NS5s, antagonize STAT2. But what's interesting is that it's species specific. So human STAT2 can be inhibited by NS5, but mouse STAT2 is resistant to NS5 antagonism. In fact, these viruses you can put in mice and nothing happens. There's no pathogenesis because the the interferon system can work very well because there's no antagonism. And so this paper is all about looking at STAT2 proteins in different species, mainly primates and rodents and bats, to get some idea about its evolution. Because, you have a viral protein, NS5, it's antagonizing STAT2. Over time, the host could change its STAT2 protein to resist NS5 antagonism, but it's not so simple, right? The host protein has to maintain its own function. And
STAT2 is really, really important. And we can't evolve as fast as the viruses can. So if you accumulate mutations in STAT2, you may not live to reproduce. And so this is a very constrained system.
So that's what they look at in this paper, the different how NS5 antagonizes different STAT2s from different species. They're going to look at Zika virus and dengue virus, NS5. And what they find is that in mammals, resistance evolved multiple times, not just once. And in some cases, it was lost. And it's complicated because you have to maintain the function of the protein as well as making this resistance. So the experimental system is quite interesting. I'll just tell you how this works. So they have cells in culture. A human embryonic kidney cells are 293 cells, which are commonly used, and they knock out the STAT gene, the STAT2 gene, using CRISPR-Cas, right? So now you have cells that don't have STAT2. These are human cells. And then they put in those cells plasmids that have a promoter that will respond to interferon, and the promoter is driving a luciferase, which you can measure easily. You can measure the light. So you add interferon to those cells, and they're going to glow if interferon signaling is happening. Of course, without STAT2, there's not going to be any signaling, so you could add all the interferon you want. They're not going to make any light whatsoever unless you put STAT2 back in, and that's what they show early on. So that's the system they're going to use. And then they could ask, if we add NS5 from different flavies into these cells, what's going to happen with interferon signaling? So you can measure it by quantifying light, right? Luciferase, and you can see the impact. Okay, that's the system that's used throughout this paper. So, for example, if you have these knockout cells and you put in human STAT2, and you find that both that Zika virus NS5 will impair interferon signaling. So NS5 is interfering with STAT2 of humans. So now you could add STAT2s from anything you want, right? Because now you have a cell with no STAT2. And what they actually do, so they want to look at murine and primate and bat STAT2, but they don't want to take the whole protein for each of these and put it in cells because you worry about differences in protein production and stability and so forth. So they just take the part of STAT2 that is the target of NS5. And they make chimeras with like bat and rodent and non-human mammal STAT2 and with the human STAT2. So they have 37 other mammalian species. And what they find is that, for example, both African and Asian Zika virus NS5 antagonize all STAT2s except camel, which is partially resistant, and koala, which is partially resistant as well. And 7 of the 9 rodent STAT2s are resistant to NS5 antagonism, as we showed before, but they find actually that two rodent STAT2s that are susceptible to NS5 antagonism. And then they also look at dengue virus, NS5, and they see a similar pattern. Rodents are broadly resistant to dengue virus, NS5, and Several non-rodent species were also, their STAT2 were also resistant, including one primate and 2 ungulates, 1 carnivore, 1 bat, and 1 marsupial. Now, I should point out that these cells have resistance, but we don't know what virus led to the resistance of these species. We're using Zika and dengue as a probe, but this evolution arose many, many years ago, and it could be some other related flavivirus that selected for that resistance.
It is quite likely an extinct flavivirus, I would say.
Yeah, I would guess. Yeah. In fact, they say for many of these species, we don't even know if they are reservoirs in nature for flaviviruses, but maybe now that we find resistance, we should check.
That is actually one of the coolest things to come out of this paper, is that you've got something to look at to identify potential hosts of flaviviruses.
Right.
Potential reservoir hosts.
Reservoir hosts, exactly. Now, much of the paper now dives into dissecting the resistance here. And I'm gonna summarize this because I think it's too much to go through individual.
And this paper, by the way,
results.
is open access. So if anybody wants the details, they can
Right.
go and download it.
So they look at the mouse resistance to Zika NS5. They narrow it down to a 12 amino acid sequence of STAT2, and they can swap this back and forth between human and mouse and swap the phenotype. But interestingly, individual amino acid swaps are not informative. They say that it's more complicated than just a single or a couple of amino acids. And they say this probably means that there are barriers to fitness for STAT2 to evolve to resistance, especially so the human STAT2, which is not resistance. It's hard to make it resistant using mouse sequences because you're probably messing with the function of the protein. And so that's one of the key findings of this paper.
Evolution is not a job for amateurs.
Yeah, it's hard. It just doesn't happen like that, and it takes time, and it's multi-step and so forth. And they say, this is very interesting, amino acid changes would need to be acquired in a specific order that avoids STAT2 variants that cannot signal. And I commend the authors for saying amino acid substitutions and not amino acid mutation.
Yes.
Good job. You must listen to TWiV.
Yes.
Actually,
pretty sure at least one of them does.
Matt Evans got his PhD at Columbia. Were you coincident with Matt Allen?
That name sounds familiar.
He was in Steve Goff's lab, but
yeah,
I was on his committee, so I may have told him multiple times, Matt, that's an amino acid change, not a mutation, because I can be annoying like that, So the other issue here that's very interesting is that so STAT2 needs to bind another cell protein called IRF9, which is part of the signaling process for turning on interferon-stimulated genes. And some of the changes that make STAT2 resistant to NS5 screw up IRF9 binding. So those are not viable in the long term. That's one of the constraints that we know about in terms of evolution of STAT2. They look at a variety of different rodent species. They find 2 that are actually sensitive to antagonism. And those are very diverged from the other rodents. So that's an interesting finding, And they study that, in some detail, and they conclude that resistance to NS5 was probably acquired at least twice in rodent evolution. So what does that mean? Why didn't it get acquired once a long time ago and then propagate through all the rodents? Well, probably didn't work for all rodents, right? It was,
and there may have been, we're talking about a long evolutionary history here. There may have been a time in the rodent lineage where one lineage was being infected by a flavivirus and over millions of years developed resistance to it, but other lineages were not affected. And then those other lineages later encountered a flavivirus and developed a similar solution, but independently.
Yep. Dixon, I thought you'd like this paper because you like flaviviruses.
I do.
All right. So then they look at
Code yellow.
prosimian STAT2 proteins like lemurs. So the lemur, one lemur, Propithecus cockerelli, the only primate whose STAT2 showed resistance to dengue virus NS5. The others are sensitive, and they say this is striking. They were surprised because non-human primates are essential to the sylvatic cycle of dengue virus, right?
Yeah.
So in other words, there's a reservoir of dengue viruses in non-human primates, and those are essential in order to provide a reservoir via mosquitoes to infect humans.
And the sylvatic cycle is where wildlife is infecting wildlife in the woods,
right,
compared to the urban cycle, which typically involves humans.
You got it.
So they dissect this lemur NS5 resistance. They find that it arose early in lemur evolution, and then it was subsequently lost in other lemur lineages that arose after this Propithecus cockerelli, which again probably was not compatible with those other lineages.
Well, and this is really interesting because it shows you can get this and then lose it.
Yep.
And it suggests that the adaptation that's happening in STAT2 has some kind of cost to it.
Right.
So, as we said, if you break STAT2, if you really muck it up, then you're not going to live. But if you've changed some amino acids to resist the flaviviruses, there's probably some kind of trade-off. It doesn't work as well at some of its functions as it used to. And so this business of the lemurs then losing this adaptation suggests that there was some advantage of not having that if you don't need it.
They also find in studying this lemur STAT2 that you can't introduce individual amino acid changes. They say they probably had to have been acquired evolutionarily in a specific sequence.
Yes.
Because if you put one in, it's the wrong one, it's going to be lethal. And so if you need, say, two, one of those two had to come first and then the second one.
Same as what they saw before with looking at the other ones.
That's right.
Yeah.
And then they find some evidence for positive selection. Now, positive selection means over evolutionary time, a particular amino acid change arises and then remains through subsequent lineages. And so they say that's evidence that a virus is putting pressure on the host to maintain that resistance. But it's not the case for all of the species that they look at. There's not always evidence for positive selection. So that's the story that basically, evolution of resistance to NS5, multiple amino acid residues, not just one. They have to. There's this idea of fitness that Alan mentioned. You have to maintain fitness of the STAT2 protein. It just make anything that makes resistance to NS5, and they have to be acquired in a specific order. And the fitness may involve interactions with protein like IRF9 and maybe other proteins that we don't even know about. Once you have resistance, it doesn't mean you have it forever. The lemurs got it and lost it. And so that probably, again, is, as they say, to avoid breaking STAT2. And then finally, in the discussion, they talk about what viruses exerted the evolutionary pressure are unknown. Right? They looked at 3 diverse flaviviruses in this study, but they say they're probably related, extinct flaviviruses. And as we mentioned before, would be interesting to go and look and see if these are reservoir hosts for flaviviruses, especially in light of increasing geographic range of vectors because of climate change. That is a problem, and so you want to make sure you're aware of what's going on. And so that would involve wildlife sampling. It would involve experiments that some people are going to think are dangerous. On the live stream the other night, someone mentioned that some people think we shouldn't do any sampling because it's too dangerous and just be surprised when new viruses arise.
No spoilers. We want to be surprised.
That's right.
Hey, we'll just make a vaccine in response. I don't think that's a good
That's not a great plan.
plan. I mean,
No.
it worked for some years, but we can do better. Isn't that the whole point?
Yeah.
Do better?
Yeah, we would rather see it coming.
All right.
If you're driving through, a snowstorm and you're worried you might wreck, turning off your headlights is not going to help.
No.
All right. Our second paper is a science research article. And this is very, very cool. Now, this is not virology, but it's about prion diseases.
Prions are kind of honorary viruses.
They're honorary viruses. Brainwide silencing of prion protein by adeno-associated virus-mediated delivery of an engineered, compact, epigenetic editor. Neumann, Bertozzi, Wu, Sirac, Harvey, Brower, Pertley, Coffey, Howard, Kamath, Lenz, Guzman, Raymond, Khalil, Deverman, Minikel, Valab, and Weissman from the Whitehead Institute, the Broad Institute of MIT and Harvard, Boston Universities, Harvard University, Massachusetts General Hospital, and MIT.
It's an entirely Boston paper.
Wow.
It is.
And the first two authors, Newman and Bertozzi, are co-first authors.
Yeah, as soon as I read that first couple of words of the title, I thought, gee, maybe we could apply this to politicians. But then it faded out really fast after that. No. So it's a fascinating article.
Prions are proteins that cause prion diseases. Prion diseases are fatal neurological degenerative diseases that are caused when one of your proteins, the prion protein, PRP, misfolds and causes aggregates in your central nervous system. And that can happen spontaneously. You can get it folding on its own. It could be genetically mediated. You could inherit mutations in the gene for PRP, and those cause misfolding. Or it can be infectious. You could eat beef from a cow with mad cow disease, which is a prion disease, and you can acquire a what they're called, they're spongiform encephalopathies. So these are a real problem because they're 100% fatal, and we have no way of treating them.
And these are really, really hard to study. They were originally called slow viruses because they were thought to be viruses, and now we know they're proteins. Right. And they take years or decades to develop, and even in model organisms, it's very difficult to, or has been very difficult to develop models that you can study in a reasonable amount of time.
And of course, Stanley Prusiner received a Nobel Prize for his work on prions for years. He said it's a protein, and people said, nah, there's a virus hidden in there. You're just missing it. There's some nucleic acid, blah, blah, blah. And he stuck to it. And the key experiment, well, he purified the protein, and he showed by inoculating it into animals, it would cause disease. And people said, there's just some viral RNA or DNA hiding in there. So then he identified the gene, knocked it out in mice, and showed that those mice are now resistant to being infected with prion protein. So prion protein gene is essential for the pathogenesis. And, in mice, you knock out prion protein gene and the mice can't be infected. They're resistant to prion diseases, but we're not going to do that in people.
Well, and a strange thing about this is when you knock this out in mice, they're, I believe, basically okay.
They're fine. It seems to be a non-essential gene.
You've got it's got to be doing something, but apparently in the brain and in the adult, and well, I guess if it's a knockout throughout development, you can get rid of this and not, at least in the laboratory, not notice any problems.
They can't do any higher math after that.
Right. If you take them to the opera, they can't distinguish Mozart from Wagner.
Exactly.
So previous studies have shown that if you reduce the amount of prion protein in the brain, you can reverse disease. You could stop disease progression and reverse the symptoms.
Right, because this is fundamentally a polymerization disease. What you need to stop is the formation of these damaging clumps of prion protein, and if there's less in there, then you don't get the formation of the clumps.
So previously, people have shown that if you give mice antisense oligonucleotides that target the prion mRNA, it will decrease production of the protein and reverse symptoms. But they say these aren't very effective, and we want to develop something better. So that's what this paper is all
And
about.
also, you have to inject them directly into the nervous system.
Yeah, you don't want to have to do that. And by the way, there are other diseases like Parkinson's, Alzheimer's, Huntington's. They involve protein aggregation
Right.
and could have benefit from some kind of therapy that reduces the essential protein. So this could have a really broad effect. So what they do here is to work on, they're not going to knock the gene out. They're going to use epigenetic silencing. Now, genes are transcribed by RNA polymerase in cells to make mRNA. It's a process called transcription. And the transcription begins at a promoter region in the gene. And the promoter area, the DNA methylation state can regulate transcription. So DNA methylation, the addition of a methyl residue to histones or DNA in this case, can regulate transcription and therefore decrease the amount of protein that's being made. So they talk about the components of the methylation machinery in the cells, and they produce methylation that's heritable, so it propagates throughout the DNA. And this has been done before using Cas9, which is a protein that can be directed to DNA using a guide RNA. And previously, people have made nuclease deficient Cas9, so it doesn't cut the DNA, but just goes there and brings a methylase with it and methylates the DNA. And so they're going to explore these approaches in this paper. And it really is a wonderful how their approach evolves as they encounter issues and they try different things. So first question is, can we silence the human PRNP gene with epigenetic silencing using a process called CRISPR-off? So they take human cells, again, 293Ts, the same as from the previous paper, and they put in them a single guide RNA targeting the transcription start site of the PRNP gene. So now that guide RNA is going to bind around the promoter, and then it's going to induce, it's going to silence it basically.
Right.
And they noticed with a pulse of CRISPR-off, they can silence the gene for at least 50 days. And this CRISPR-off, by the way, it has Cas9 in it, and what's the other component? I think it's bringing in the methylase as well.
Yeah,
right.
I think so.
CRISPR-off. Yeah, I believe it's bringing in the methylase, and it's targeted to the promoter by virtue of the guide RNA, which Cas9 brings in. So this works. You can turn off the gene by doing this approach. They also try another neural cell type, and it works there as well. They use a mouse cell type, so they say, we can use mice to assess this. Okay, but they say we don't really like CRISPR for prion diseases. We'd rather use an adeno-associated vector.
Right. So an issue with CRISPR is it's a large, complicated system to get into the brain. And adeno-associated viruses are a very well-characterized gene therapy vector that's been used in lots of tests and trials, and is much more effective at delivering payloads to the brain. But you can't put this CRISPR-off system into it because AAV is a small virus that can't accommodate it. all
Right.
this
It's a
machinery.
commuter airline rather than a 747.
Yes, it's almost like a Cessna 152. It is
There
very
you go.
compact.
There
It
you
is
go. There you go. Or
Yeah,
even a
so
glider, perhaps.
yeah, and you could potentially, they talk about the possibility of including, it was either in this or the perspective that accompanied it, both of which unfortunately are paywalled, so sorry. But you could introduce components in different individual viruses, but then you have the problem of making sure that they all actually get into an individual cell. It
Yeah,
becomes a lot
right.
less efficient.
Did you point out the advantage of AAV as it pertains to getting into the brain? Does it actually go through the choroid plexus?
Yeah, so they've actually engineered AAVs that have improved brain entry. In fact, you can make AAVs that'll get into any tissue that you want. It's really amazing.
Is that a gain of function?
It certainly is.
Yeah. So if you ban gain of function, forget about all these therapies.
Exactly.
Right?
Exactly.
Which could save many lives, right? I mean, just
And probably
I think it's about definitions.
prion
Yes.
diseases are not that frequent, but certainly Alzheimer's and Huntington's and Parkinson's would be. Right. So as Alan said, the Cas9 is too big. So, however, we do have other DNA binding proteins, and one of them are zinc finger proteins.
Uh-huh.
And you can program them to have specific genome target. They're small, they're human proteins, so they're not going to be immunogenic, whereas the Cas9 is a bacterial protein.
Yeah, so this is the zinc finger is the binding domain that's found in many DNA binding proteins, and you can engineer just the zinc finger, which is a very small piece, as Vincent said, to target a specific thing, and then you could potentially attach something else to this targeting zinc finger.
Right. And so they come up with one approach is to put the methylase in the vector along with the zinc finger, which would be okay, but they find that the methylase is toxic on its own, so overproduction. So then they say, all right, let's not put the methylase in, but let's put in a protein that will recruit the methylase. So we'll use the DNA methylase that's produced in the cell, and we'll just have zinc finger next to a protein that will bind the methylase.
Why pack your own methylase when you can just buy one on the spot? Yep.
It's just, and that protein is called D3L. And so they use that and they show that works. And in fact, they further modify it by adding a little histone tail that will further recruit the methylation machinery. And they show that works even better. And they do a model system where they have cells making a fluorescent protein, and they target the promoter of the fluorescent protein to try and silence it, right? Because that's a very rapid assay. And they can try different things, and they end up making this construct, which just has, well, initially they do Cas9 linked to this D3L protein. And they show that works, and they can optimize it and get various parameters to work. But then they say, let's get rid of Cas9 and put the zinc finger binding domain. So they replace it. There's a published zinc finger domain that will target the mouse PRNP promoter. They put that into their construct. And there are also other proteins other than zinc fingers that work. Another one is called tails, transcription activator like effectors, which they also test and they show works as well. Here's another thing they test is making. So there's a phenomenon called transprotein splicing in eukaryotic cells. So people may know that mRNA can be spliced by removal of sequences that are not needed. Well, proteins can splice themselves together. It's called trans-splicing, and they make vectors that will make two components of their system and then get spliced together, having the right sequence in it. So you can even reduce the cargo.
This is so
burden.
Clever. It reminds me of object-oriented programming on a computer where you have these individual pieces that you can assemble into various ways, and well, I say this frequently about papers that we cover, but this is a boatload of work.
They've gone through
the amount that's in this paper is just
huge,
staggering.
Look at the authors.
They built
authors.
Sorry,
you've got 13 authors.
It's still, that's not a lot of authors for the amount of work here.
Look where they are, though. That's the point.
Yeah, and so they've got this system, and they can build it in a modular fashion, and that allows them to save space in this compact AAV genome, so that they can do even more with it.
Right.
I mean, it all builds on fundamental molecular biology that's been learned in the past 50 years, right? The vector science, the CRISPR-Cas9, the zinc fingers, the protein splicing, all of that basic stuff, and now they're using it.
Yeah.
So, and I think a lot of that basic science was discovered serendipitously, right? People didn't say, we're going to go discover protein splicing. They found it.
Yeah.
So this is an argument for letting people
basic
follow
research,
their curiosity.
That's
Yes.
right. Stay curious.
This is what you get in the end.
That's it. That's right.
Okay, so then they make they make lentiviruses with this system targeting PRNP. The silencing is specific. There's very little off-target silencing because the zinc finger is specific for the PRNP gene. And so now they said, what about the brain? Let's try it in the mouse brain. So they make their AAV viruses with this system, which they call CHARM, by the way. And I have to go back to where they define what.
Oh, yeah.
It's
a cute little acronym they came up with.
It is
coupled histone tail for auto inhibition release of methyltransferase.
Thank you.
CHARM.
Exactly right.
That was my contribution to this show, by the way.
So they use a capsid of AAV engineered for high transduction throughout the mouse CNS. You asked about that before, Dixon.
Yeah.
They give intravenously.
Yeah.
Right? So that's the cool part. 1 × 10^13 viral genomes per kilogram to adult mice via retroorbital injections. And then 6 weeks later, they harvest the brains and they do RT-PCR to measure the PRNP. 70 to 90% decrease in PRNP transcripts and a 60 to 80% reduction in PRP protein levels. Amazing.
Yes.
They play with the doses and they find they can put. So basically what they're done here, there's no DNA methylase in the AAV. It's this protein that's recruiting the DNA methylase, and it's targeting the PRNP promoter by virtue of the zinc finger domain.
Remarkable.
They also do in situ hybridization to see, because remember, they just ground up the brain in the previous study. They said, where are these neurons? And basically the silencing happens throughout the brain in most neurons. So it propagates throughout the brain. These are post-mitotic cells. They don't divide, so it works in them. Okay. So maybe you're worried about this being on all the time, right? Turning off. So let's put an off switch in it.
Because why not? I mean, you're putting in all these features. Why not add another?
Exactly.
They add a zinc finger that will bind the promoter of the virus that's driving the synthesis of this CHARM cassette. So we silence their own promoter after silencing the target. And I love how they write, we installed the zinc finger motif.
That's right. It's like a mechanic and working underneath a car.
We installed it.
Yeah, and this is so cute because you've got a system that you now know can turn off a gene by hypermethylating it. And so you just include another piece that will turn off the gene that you just put in that turns off genes.
That's right.
And so it'll turn itself off after it gets settled in.
So you can go on vacation without worrying about having turned off the light or not.
Yes, exactly. It's on a timer.
It is on a timer. That's right.
So they make a variety of constructs, and what they find is that even with the self-silencing, they induce repression, but the ones with the self-silencing, after a certain amount of time, the repression is gone. So they think it probably got repressed too quickly. The vector is. So they play a lot with trying to tinker with that, and they find one that is self-silenced and has stable repression over time, and that's the one that they go forward with. And they use those in mice. They package them in AAVs. Again, PRNP expression strongly reduced 6 weeks after AAV infection, and no evidence of reactivation even when the vector is inactivated itself. And they say we still need to do more optimization to balance the self-silencing kinetics with silencing the PRNP gene. But they have amazing ways of doing it. They can change sequences in the various proteins that are part of this vector, and I just think it's amazing what they have done here.
This is just so cool.
So it takes the existing DNA methylation machinery and brings it to your promoter of choice. So you could pick any gene and just use a zinc finger protein that targets that gene, and you could silence it.
Yeah.
Right.
And you can, because of the compactness of this, you can include multiple targets in
That's right.
one virus, and you can have this shutoff system built into the virus so that it acts for a limited time.
Yeah, so I would refer to this as molecular duct tape.
It's more sophisticated than
I think
that.
it's very sophisticated. Yeah.
This is a timer.
It has its charm. It has its charm.
It has its charm.
No,
yes.
it does have its charm, doesn't it?
That's right.
This is the first AAV-delivered tool capable of gene silencing through targeted DNA methylation.
Right. So I'm sitting here, not necessarily silenced, but trying to recreate how you acquire prions, and you acquire them, as far as I know, by eating meat.
One way is eating.
One way.
Okay, so that's a protein that you're eating, and the protein resists digestion.
Yes.
Then it gets into your blood. Then it goes into the brain. Then what?
Then it causes your prion protein to misfold.
Oh, you have a prion protein. Okay.
Yes, we all have normal
we all
prion
have prion protein in our brains.
Okay, that's opened the door.
And once it causes your prion protein to misfold, then that misfolded one can cause other prion proteins in your brain to misfold, and it's a chain reaction.
Yeah, I got it. So we have no other name for our prion protein.
Prion protein.
Prion protein, that's it.
Do we know what it does?
Nope. Apparently you don't need it, though.
So apparently you mentioned that you can silence it in mice to begin with, and nothing bad happens except that they max out their credit cards or something like that.
Yeah, as far as we can tell, nothing bad has happened to the mice. But
you have to speak mouse in order to find out really what's going on.
Exactly, right. Maybe they can't speak French anymore.
Well, so in mice, you could actually knock it out, and the mice seem fine. But we're not going to knock it out in humans, because knocking out genes, there are off-target effects. You may knock something else out.
Understood.
CRISPR-Cas is very dicey in that sense.
Innocent bystander
just
effects.
Methylating is beautiful, and they've shown that it's specific for the promoter because of the zinc finger protein. Right?
Yeah. Got it.
So you could any protein that is produced that's misfolded or wrong, you could turn it off. Right?
Yep.
It's really cool. Now, for example, one of the treatments for hemophilia is to replace the mutant copy with the wild type, but I would say you just silence it and add a new one in. You don't have to do that gene replacement thing.
Yeah.
And it's a long-term process too, by the way, to cure hemophilia. It's not straightforward.
Right.
It's a long-term therapy that's extremely expensive.
Yep.
Now, people may ask, okay, how do we know that reducing the protein is going to make a difference? Well, in mouse models, it has made a difference, but we don't know in people. So we have to do clinical trials. But first, we'd have to have a product that we think is the final candidate. You have to do some animal studies.
There's more optimization that needs to occur here. There are more animal studies. You need to look at it in more species. And you need to do very, very thorough analysis to make sure that turning down prion protein doesn't have consequences that we would worry about.
Right.
And it's important to note, prions, I mean, you mentioned that this is a similar pathogenic mechanism to things like Alzheimer's disease and Parkinson's, which are much, much, much more common. So why not treat those? Prions are a great target as far as clinical trials go, because we know that when people start developing the symptoms of these, it is 100% fatal.
Mm-hmm.
And so if you go into people who have this 100% fatal, rapidly neurodegenerative state going on, you're a little less concerned, and they're probably a little less concerned about, well, we don't know all the side effects. Whereas somebody with early stage Alzheimer's disease, if you say that, and well, maybe I'll wait till you're a little more sure of the side effects before.
So if you're interested in numbers, worldwide, the rate of these prion diseases in humans is about 1 per million. So in the U.S., we get about 350 a year. So that would be the basis for your clinical trial.
Yeah.
I guess you could do it internationally and get more people as well.
Yes.
Right.
Yeah, they would have to go outside of Boston.
You bet.
It's very cool. I just think this is not only a medically important topic, obviously, but broad implications for other diseases.
Yeah.
As well.
Yeah, and this is, I'm sure they've submitted patents that are not specifically to AAV and all the specifics of this, because you could use this in a much wider range of ways.
I wonder if you could silence the HIV promoter in people with HIV/AIDS permanently.
That would be pretty sweet.
Permanently, right?
Yeah.
So you wouldn't have to take your antivirals your whole life.
Yeah.
Roger that.
It's a great idea.
Nobel Prize winning idea, I'd say.
Yeah.
I don't know about that, but
oh no, if you got rid of AIDS, we would be calling you sir.
Well, I'm not going to do it, but someone else should do it.
Someone else should.
Sure.
I'm sure somebody else will look into that.
Yeah. Very interesting. Okay. Let's do some email.
You bet.
All right. First one. Alan, can you take that one?
Sure. Miffy writes, "Hi, TWivers. Thanks for deepening my understanding of virology. I've been listening to you for a long time, since way before the pandemic. I remember when I started hearing the first whispers of a mystery illness in Wuhan, I thought, whoa, that podcast is going to have a spike in listenership. While I am a loyal listener, I am perhaps not among your target listenership. That is to say, I am not a scientist of any derivation. Rather, I am just an arts grad and a career writer and editor. I now work in communications at a Canadian top 5 post-secondary research institution, but it is not in that capacity from which I write to you. Since I am not a virologist nor any kind of scientist, maybe my next comments are not in scope for the purposes of your podcast, yet here I go. I will admit that sometimes I am rubbed the wrong way by some of your comments. Like in the first 10 or so minutes of episode 1125, you rightly criticize as partisan the efforts of Republican lawmakers at the Energy and Commerce Committee to restrict the work of the NIH as reported in Science. But then you go further, wondering how any non-scientists dare form opinions of scientific work and how it's carried out. My friends, I am here to tell you that it is literally the job of lawmakers and elected officials to make laws and govern in ways seen as in the best interest of society, even when people disagree with them. The Energy and Commerce Committee's name is inapt, but its jurisdiction, particularly that of its Subcommittee on Health, is clear. You and I may be free to vote otherwise, actually not me, I'm Canadian, so I can't vote there, and we may be free to publicly opine and decry the committee's efforts as partisan, but it's patently ridiculous to assert that only scientists ought to have a say over how science is carried out and over who has the exclusive right to be the arbiter of what is acceptable scientific work. That kind of gatekeeping is off-putting for us ordinary unwashed masses of non-scientists. People talk all the time about practices in, say, for example, education, media, or medicine without being teachers, journalists, or doctors. Further, in addition to industries having their own professional regulatory bodies, we have public policies and laws about all aspects of society exactly because no pursuit exists wholly apart from society. At one point in the episode, someone even cracks a joke about gain-of-function research in China that at best is confusing and at worst mocks listeners and undermines the careful work the podcast has done to refute theories of coronaviruses being leaked from labs in China. Maybe the podcast is only for scientists? I am a dedicated TWiV listener, but I'm just an arts grad. Have I outstayed my welcome? No, Mifi, you have not outstayed your welcome, and thank you very much for this little nudge.
Indeed.
Which we should, I think, should take to heart.
Yes.
That there are some very good points in here.
I do think that, as we mentioned earlier, if lawmakers are going to regulate science, they need to know what they're talking about. So you can't just say ban gain-of-function research, because that's absurd.
Yes.
Yet that's what they're doing. And that's why we sometimes talk this way. And I understand that lawmakers have to make laws. I get it. I totally get it. But I think you need to know what you're talking about, right? Now, doctors do things and are regulated by non-doctors, but the expertise lies in the doctor, and it has to be appropriate for whatever is regulated, right? The people who regulate airplanes, Alan, it's
they are not all pilots.
They're not all pilots, but pilots are amongst them, right?
Yes,
that's right.
Yeah, so I think in terms of science regulation, the distinction here is on questions of fact, the scientists have a perspective that is unique and that needs to be taken into account. And if a policy is going to be too vaguely worded to allow any science, so if you throw around the term gain of function and you don't very clearly define what you're talking about, as we've said on the show, that would encompass an enormous amount of science that I'm quite certain even the most extreme Republicans in Congress don't want to ban. So they do need to understand the fact side. On the other hand, when it comes to questions of public policy and funding the research that we're talking about, it is totally within the right and job of Congress and other elected officials to say, my constituents aren't comfortable with funding certain types of research, so we're not going to fund those specifically defined categories. The scientists can then explain how you might define the categories that specifically offend you, and you can proceed from there. Because it is this congressional process that's paying for the science. We do need to remember that. But yeah, I mean, Vincent, you have an excellent point. They can't just throw out terms and think that that's a specific term of art when it's not.
No, and I would add that Vincent's complaint is also directed at the center, the core of why we became scientists, because it's fill in the blank. Okay, I wonder what would happen if I. That's the question that all of us are always asking, and we are curious people to the point of having to create P4 facilities in order to answer some of them, which is a multi-billion-dollar extravaganza in some people's eyes, but in others, it could save millions of lives if the right things are discovered by filling in that little blank. I wonder what would happen if I. And preventing that question from being asked is tantamount to telling somebody that you might as well do something else for a living because, yeah, that's what brought me to the lab to begin with. I wonder what happens if I. And so,
as we saw in that last paper, when people get curious, things happen.
Yeah.
And that's what I don't want to squelch, right? That
No,
curiosity.
That's what I'm saying. And
by
so,
regulation.
What I would suggest, I know this is difficult and it would create a clumsy system in some ways, but whenever that question arises that you might be stepping on the toes of science in general, why don't you invite some scientists in to explain it in common terms so that you can understand the ramifications of that law that you're trying to pass?
That is how the process is supposed to work.
Well, I know that, but that's not how it is working.
Well, that is a separate problem that has to do with the current state of politics in Washington.
Well,
And I don't have a solution, scientific or otherwise,
yeah,
for
but
that.
this woman is in Canada. I don't know if that happens up there.
Right, she's writing from Canada, but we were talking about the U.S. situation.
Sure.
But Maifi, very good points and well taken.
So that I get very emotional about this.
Yes.
Because I have seen how science works at its best.
Yeah,
and any attempts to change it, especially uninformed attempts, really make me bristle. So I'm sorry about that, but I'm very passionate about it. The last thing I want to say is it makes zero sense. I understand that lawmakers should regulate, but it makes zero sense when one party is doing all the regulating and not the other. Okay? So, and we know what we're talking about here. The politicization of science, it makes no sense. We can't do that. Okay.
Okay, then.
Alan, you took it. Dixon, take the next one, please.
Moniqua and Noah, right? Hello. I hope all is well. This past week, Noah completed all aspects of the fundraiser from last month. $2,800 was raised for 5 programs, and over 15 girls were able to attend a STEM camp. With the funds from raising donations, $560, a portion was given back to Noah as STEM boxes from the amazing subscription program called Girls Can Create.
No, crate.
Crate, I know, but I'm saying it the way they wanted it. Girls Can Create, and that's what they would like you to say, because that's Girls Can Create.
Well, I think it's the girls. Crate. I think since it's a box, I think it's the girls can ! Crate.
Yes, crate. That's right. But if you say it fast.
Anyway, continue, Dixon.
I will. At least that's the way I interpret it.
Okay.
These boxes were distributed by 2 facilities that focused on children in foster-based programs. We wanted to let you all know how much each of your programs has meant to both of us, and how proud and grateful for all you do for children. Noah hopes that the funds raised have helped in a small way to expand our help where needed. Hope next year will be bigger and more programs added. My daughter hates attention, but understands the need for others to see members of the community helping those in the community and beyond. Below is a quick, proud mom picture. Have a wonderful summer, and again, thank you.
So Monica and Noah had written some time ago asking us to mention their fundraiser, and we did. So I'm happy that it worked out.
Excellent.
So Girls Can Create, basically they deliver a toolkit filled with information to inspire female role models, right? To show that you can be Marie Curie or Junko Tabe, you can do anything. So it's information about what women have done in these careers and how you can do it too. So she sent a picture of these boxes that they bought with some of the donations in which they're shipping out. It's very cool.
Excellent.
Yep.
Anthony writes, with respect to vaccinating pigs. So lately we've been asked, can you vaccinate pigs? Can you vaccinate cows? My understanding is that even the Kingdom of Saudi Arabia found convincing those caring for camels to vaccinate when there was no economic concern to be an intractable problem. KSA did find it easy to compel manufacturers seeking licensing to include a MERS vaccine as a component in products to protect against significant camel disease.
That's a good approach.
Yeah, we've talked about this a bit. We also talked about it Wednesday on the live stream. Just there's a lot of money we're talking about here. And just to test the vaccine, because, in the case of, say, cows infected with H5N1, you have to show that it works, any vaccine that you make. So you have to do a clinical trial.
You bet.
I don't know who's going to pay for it. All right, back to Alan.
Okay, Walt writes, in 1125, you mentioned that Congress wants to, quote, restore Congress's role in directing funding, end quote, and concurrently to create an independent review entity for the proposed National Institute on Infectious Diseases. The obvious politicization is not uniquely aimed at your field. As described in, provides a link, and elsewhere, conservatives are broadly aimed at all federal agencies' ability to carry out congressional mandates created over the last 10 decades. Quote from the article, Republican lawmakers and the conservative legal movement have been critical of the doctrine for years, describing it as a way that regulatory agencies go beyond what Congress intended when it passed laws. With the High Court's current majority of 6 justices appointed by Republican presidents, federal agencies have been met with increasing skepticism. Several justices, including Alito, Thomas, and Gorsuch, have questioned the Chevron decision. Continuing the letter, prior to a year ago, this has been primarily a pro-business anti-regulation effort, but NIAID has been added as another target thanks to Fauci's contradiction of Trump. Nicely described in this book excerpt, provides a link to an article in The Atlantic. So maybe the world does need This Week in Science and Politics. Mr. Dove would seem to be a likely expert to host it, but maybe Microbe TV simply needs to advertise another group's work in this area. Best wishes, Walt from Oakland. Yeah, and in fact, this decision, I think, just came down, and it went the way you'd expect. The Republican justices, all 6 of them, went ahead and struck down this Chevron precedent that was basically the way federal agencies were interpreting things that Congress had left vague. And so now they can't do that. And so this throws a tremendous amount of federal regulation into limbo, which was the intent from the beginning.
Not Rush limbo.
No.
Chevron doctrine.
Right. So this is a doctrine that says when Congress doesn't spell out exactly how a particular law is supposed to be implemented, it is up to the agency to provide the expertise to the regulating agency to have the expertise to figure out the details.
Right.
I'm summarizing, but people can look this up and get the gist of it. By striking that down, the case that actually caused this beef was commercial fishing boats are required to have an observer on board from from some government, I think Fisheries and Wildlife, to make sure that they're following the rules and such. And the arrangement for paying those observers who are going out riding on a fishing boat, they're not going to do this for free, has been that they charge a fee to the fishing boats, to the captains who have to pay to cover the fee of the inspector who's on board. And so the fishing industry sued over this, and this has now been overturned. And the reason this was implemented that way is because when Congress passed the law requiring the inspectors to be on board, they didn't say how those inspectors would be paid. And so this was the mechanism that was implemented. Now, instead, they have to go back to Congress and say, you need to spell out the mechanism for paying these people because you forgot to do that. So it has to go through a whole new round of legislation in order to fix that. So for a science example, if Congress decides to ban gain-of-function research, and then the agencies responsible for enforcing that, say the CDC, interpret what gain-of-function means, they would run afoul of this new decision and would then have to go back to Congress and say, you have to define exactly what gain-of-function means.
Well, presumably they would.
Yes, but this is a way, this was intended, this case was brought as a way to gum up regulation, and that's the way it was decided. So it's now more difficult for government agencies to regulate.
So what's the end game of all this, Alan?
The extremist right-wing fantasy is that we don't have government, we just have businesses and contracts, and that's that would be the way the world worked, which would end up being pretty dystopian if you've looked at any models where anything like that's been tested.
So no federal government or no local?
I don't know how. It depends on how far people believe on this spectrum, but yeah.
It's amazing a group of people that are so anti-government are so anxious to run government.
Yes.
It's kind of
well, to destroy it is the
That's right. That's where they're from.
Anyway.
Well, they want to destroy it and put themselves in place as a dictator.
That too.
Mhm.
Yes,
I mean,
there's a lot of
I don't
that
want a
going on.
freaking dictator.
Well, I must say that there are a number of amendments to the Constitution that have similar ambiguities associated with them.
Sure.
And you're running across one right now as to whether a former president has immunity from prosecution. And that has not been spelled out in ways which is clear enough to eliminate doubt.
Yeah.
So there.
If they don't
Should I
say
take the
you're
next one?
immune, shouldn't you assume you
You're
can be
not.
prosecuted?
Yeah.
That's right. That's
I mean-
exactly right. That's exactly right.
It seems to me that you're making shit up if you
There is,
do otherwise.
there is no law that says that, of course, but one can cling to their fantasies until proven otherwise.
Oh my gosh.
Craig writes, you use the old saying, ignorance is bliss. Friends, knowledge is power, quotes. But for decades, that hasn't been true. Knowledge is bliss for those of us who enjoy learning and understanding more of the world. Ignorance is power. Unconstrained, but reality, facts, or evidence, you can argue and attempt anything you want.
Apparently. Yeah, you can say vaccines have never been tested.
You can say whatever you want,
Yes.
but whether people believe you or not, that's the other thing. So I loved last night's debate, pardon me, that had facts checkers all over the place. And you can now read the facts and you can read the checks, and they're quite remarkably different.
Sarah writes, first of all, thank you for the great and informative podcast. When I discovered TWiV at the start of the pandemic, I had no idea that viruses could be so exciting. On TWiV 1125 and the plans to reform the NIH, I too believe this is a political attack on the NIH. Since science has unfortunately become political, how about a special episode to explain the overarching context of this rationally pointless measure and the overall politicization of science, for example, with the political historian Heather Cox Richardson as a guest. She's a professor at Boston College and author of the Substack Letters from an American. I think it's important to understand what is happening right now because these political decisions will have a big impact in the long term. I am familiar with Heather Cox Richardson, but I don't know if she would be the person to talk about this. I have no idea. Have you read her columns, Alan?
I have not.
I have not.
I used to a lot, but then
I
just
read as little politics as I can.
yeah, it just scared me too much, and I stopped reading it. Right. I saw an Instagram graphic today about news trends globally, like different colors for different areas. And the US is when something happens, the press blows it up and makes a big deal and you're all going to die, blah, blah, blah. And Africa is like, we don't care. We don't care about anything. And in Europe is like, I have other things to do. Very interesting.
Right.
Anyway, if you have ideas for someone who could talk about politicization of science, that would be good. I don't know of anyone.
Sure.
Well, Tony Fauci, I'm sure he's got some opinions, and now that he can render them in public, I would love to know what he thought about, all the years that he spent at NIH. He must have seen a lot of ups and downs.
I'm sure.
in terms
It'd
of
be
that.
great to have him on the show again, but I think for this particular discussion that Sarah's writing about, it would be good to have somebody who studies this in particular.
Okay, folks, listeners, let us know who that would be.
Yeah.
We're back to you, Dixon.
We are, eh?
Or is it
I don't
me?
think so. No, I think it's
It's Alan. Sorry. Yes.
I think it's me.
Alan.
Yes, it's Alan.
Anil writes, Dear TWiV, I am a big fan of the show, and I download your podcasts and listen to the episodes while commuting to work. Of course, I go through quite a few shows a week, as my commute is almost 2 hours, 95 miles one way, from southern Massachusetts to the quiet corner of Connecticut, not too far from Springfield, Massachusetts. Wow, it's a heck of a commute. I really liked your recent episode on debunking the lab leak theory, and it is sad that more people don't listen to podcasts like these instead of the angertainment of other shows. I love that term.
Angertainment.
Thank you, Anil. Angertainment. I feel your work helps counteract the slow dumbing down of society and helps elevate our knowledge, level of thinking, and appreciation for science. I am an infection preventionist, and I currently work on contract and will be switching to another location soon, hopefully without a 2-hour commute. Pardon me, had to pause to cough. We IPs can be the ID doc's right hand in the hospital, and I often find when hospitals hire the right people in the mix of controlling infectious disease, it can be far more successful than even the C-suite could imagine. Keep up the good work, and it would be my honor to meet you guys in person one day, especially Dr. Dove, who I gather is out of Springfield as well, home to America's first automobile. Yes.
Really?
It is a city of firsts. We have a whole bunch of stuff that we lay claim
Got
to.
the Basketball Hall of Fame, too.
Basketball Hall of Fame is here. Volleyball Hall of Fame, by the way, is in the next city north,
Get out.
in Holyoke.
Just over the net.
Yes, just over the net, right. So if you're into Hall of Fames, you're well served here.
I'll be darned.
Anyway, finally, I am also a big fan of electronic music and Miss Monique. Try out music from Boris Brecha and Sojo if you have the chance. Loyal fan, Anil. Thank you, Anil. Great letter.
And Dixon.
John writes, I'm not seeking to get this question on the air, but just to have you all look at the article in Reuters. I'm sure others will send it to you for review. In 6/14/24, Reuters has an article revealing the U.S. military stoked vaccine fears for Sinovax, test kits, and PPE using Twitter a few years back, and then he gives a reference, and of course we've already talked about that a little bit. It does at least state that Sinovax wasn't as effective as available options in the world, but only much later in the paper, and does not excuse the Pentagon for doing something like this if true. However, they paint the picture that the Chinese government was completely unprovoked in blaming the U.S. and creating conspiracy theories that the U.S. created the pandemic. That is the part of the article that I took issue with, however. It has been long enough. I am questioning my own memory, so maybe you can all assist. I thought first we were already straining our relationships due to geopolitical issues like new tariffs and hostile provocations, and then the former administration antagonizing the Chinese government further with calling the novel virus the China flu, among other accusations of WIV involvement. Wasn't it our politicians making unfounded accusations that caused the Chinese government to clam up and to begin firing accusations back? Or is this article correct in its portrayal of the U.S. retaliating an unprovoked China? I seem to remember that it was our politicians creating the poor relations, which didn't help our investigating team. But like I said, it has been a long time to forget the timelines of events. Anyway, this doesn't excuse a horrible action. I think, however, whoever dreamed up the policy to use our technical capability to call into question any vaccine, regardless of the purpose, is unethical and maybe even warrants calling it a war crime, since the effects seem to be children not getting the regular vaccines now that parents have bought into the fear mongering. This is a very disappointing, and if true, it continued with the current administration and didn't stop until spring of 2021. I find this all very upsetting, and I imagine that you all do too. And I would like to know the veracity of these reports, but on the surface, it looks pretty bad. John.
I think that's correct,
yeah.
right, Alan?
As we said above, this is pretty bad, and China was not unprovoked. As this virus was emerging, there was this nonsense coming out of the U.S. administration at the time about how the virus was China's fault, and we started that. And that led to the Chinese response of hunkering down and, throwing their own accusations, which also, got out of control, and they've come up with all of their own conspiracy theories. But, we were not innocent in that debacle.
And I believe, though, that there is some history here with regards to the original SARS outbreak in China, which the Chinese government tried to keep silent.
Yes.
Because it inhibited about 65 million tourists from coming to China. And and so that kind of history sort of, well, it's happening again type of attitude, whereas it might not have been, but certainly at that point it appeared to be. In fact, I was part of a team that came over from our School of Public Health to go to China to sort of consult with their Chinese counterparts in public health to see what could be done about improving communications between uh, infectious disease people, the governments, and the public. And
Yeah.
you'll never guess what happened after
What
we
happened?
left. 3 weeks after we left, China's government fired the entire public health department
Hmm.
and restaffed it with other people.
Well, you know, in SARS-1, the market remained open for 7 months, and
Yeah.
they didn't tell the rest of the world for many, many months.
That's right. That is right. That was in Guangzhou, as I recall.
Yes,
Yeah.
it was.
That's right.
Okay, time for
Crazy
some
stuff.
picks of the week.
Yep.
Dixon, what do you have for us?
Well, I have another space odyssey, so to speak. I didn't know Richard would not be here, but I'm glad that Alan is. This is a flying monument to our technological capacity. We have a Mars Odyssey orbiter, which is going around photographing the surface of Mars. Whenever you want to relax, just go to the website and put the big pictures of an inside of a volcano or a crater, a desert, the polar caps. But nothing looks Earth-like. Nothing. It is remarkably different in every aspect that I can tell you about. And it's relaxing to just look at these wonderful patterns of nature expressed in another place. This Mars Odyssey orbiter is about to make its 100,000th orbit. And I think that's worthy of note. So I picked that as my pick of the week.
This is really an impressive accomplishment. The thing has been in orbit for 23 years.
Exactly.
Around Mars and just keeps
Exactly.
on relaying data and terabits of data
You got
and
it.
just
Well, Voyager
provides
1,
so much information.
yeah, Voyager 1 takes the cake, I think.
Well,
Yes.
for distance, yes.
Yeah.
Yes, that is.
And they did get it to actually reactivate,
Yeah.
which is, I can't believe that a radio signal could go that far, that fast, but there you go.
Yep. It's
This
at the
is what-
speed of light and it just keeps on
I
going.
love it.
This is what happens when you give scientists money and let them do their
You're
thing.
right. No, Vincent, you are on the
Nobody
money.
tells NASA what, yeah, we're going to explore.
You're on the money.
That's it. Of course, in this case, there's no danger involved, right?
Yeah, right.
Well, every launch has risk.
For sure.
Pieces can fall and stuff can go wrong.
and they do. And
yeah.
by the way, we've got 3 astronauts up in the International Space Station waiting to come home. And the Boeing Starliner, I believe
Their
it's
flight
called,
has been delayed.
It's a little bit delayed. That's right.
Yeah.
So there's a switch on the tracks that didn't engage or something. I don't know. That's why the subways are late in New York. So
Their boarding pass system.
the
got hung
boarding
up.
passes weren't approved. That's right.
So
Their baggage was overweight.
they're waiting for a rocket to come get them.
Yeah, they are.
And what happened to this Boeing thingy?
Well, they've got the satellite. They've got the capsule sitting up there.
Yep.
And nobody knows why. There are several glitches in it, which would not guaranteed blow up, but the possibility is there. So the odds are great that you would survive and everything would be fine, but they don't want to come home under those circumstances. So they may have to send
So let me
another
understand.
spacecraft up for
So they're going to launch a capsule to go dock on the space
They
station.
might.
And bring them
they
back?
did already.
I think at this point they're still troubleshooting the Boeing capsule.
Yeah, they want that to work in the worst
And
possible
they
way.
have other capsules that are scheduled to launch and go up to the station anyway. So
That's right.
It's not like it would be a special, we got to build a rocket to go get our astronauts back. This is something that they're having some issues with this particular capsule. And, it's not like Boeing was in need of any good press or
That's right.
anything.
They've been through a lot
So
lately,
before-
haven't they?
Then we used to come back in Soyuz
Yeah.
vehicles, right?
Sure.
Well, no, but did other capsules also.
Americans, U.S.
Yeah.
Oh, yeah.
I thought we had to come down in Russian ones.
Privately.
We had. I think we've had our astronauts go up and come down in Russian
Yeah.
ships, and Russians have gone up and down in American ships, and
Right.
so there's a lot of international collaboration with that.
That's right. That's right.
But they've got high standards for safety, and
They do.
you know, if things don't check out before the flight, then you don't launch until you can make them check
Right.
out.
They've been down that road before.
All right, so
they
let me
don't wanna see that
understand this because
happen.
I don't follow space. So there's no more NASA.
Oh no, there's tons.
Oh,
of
there's
why-
NASA.
But they're
What's Boeing doing?
subcontracting.
Did they make it as a subcontractor for
Yeah,
Yes,
NASA?
they did.
Boeing is a
They-
contractor. Boeing and SpaceX both contract with NASA.
I
Many,
see.
many other companies also contract with NASA, but you've heard of Boeing and SpaceX because they do the launch vehicles.
Got it.
Right.
So this is what's going on here.
Exactly.
So this is a
And the
change
other-
because NASA used to build its own shuttles, right?
The brilliant.
They
part of
still
this-
worked with contractors. The shuttles were built, all the space vehicles have been built by companies
Yeah.
that were hired by NASA to build components of the space vehicles.
Right.
And now we actually have companies that are putting entire launch programs into place for a variety of reasons, not just for the space station, but because they want to have satellites and they want to have, you know, all that other stuff going
Yep.
on.
But
Okay.
there's an economic incentive for them to do it also. The people that sent the Boeing capsule up with the astronauts recovered both rockets that were fused onto the sides of the large rocket that continued up to the space station. They actually landed back on a platform
Hmm.
where they were launched from without incident.
Yeah, that's the SpaceX thing.
And they've saved, I don't know how many billions of dollars just from those two rocket units that used to fall into the
The
Atlantic Ocean
reusable
and get
rockets,
lost.
yeah, that has been a great
So
advance.
that's
Sometimes
been the
they-
big incentive for privatization.
Sometimes they
is-
blow up, though.
Sometimes
Yes.
they do blow up.
I've seen videos of they land and.
Oh
Yeah.
yeah, well that's exactly right. So you
Not
get
funny,
it right.
but they're unmanned. Okay, I've learned a bit about space. Alan, what do you have for us?
I have, so my actual link this week is an example of a type of institution. This happens to be a link to my local animal shelter. But I recommend that people think about their own local animal shelters in this time of year. If you happen to be anywhere near Western Massachusetts, the one I'm linking to is a great choice. But anywhere else in pretty much the whole Northern Hemisphere, there's something going on. Happens every year. It's called kitten season, which sounds adorable and to some extent is, but it is also an immense logistical challenge for animal shelters all around the world. And what happens is cats have an estrus cycle. The female cats come into heat every spring if they're not fixed, and the unfixed male cats are very diligent about ensuring that any female cat in heat will become pregnant immediately thereafter. So what happens is that all the stray cats and all the unfixed domestic cats start reproducing like crazy, and they will produce 2 or more litters in the course of the summer. They are now inundating your local animal shelter wherever you are. If you have any inclination to adopt a cat, now is the time to go do that. So that's my little plug for adopting animals, because now's a great time.
What is the ethics of spaying all those cats?
They should all be spayed and neutered unless somebody has a very specific plan. Actually, even if you think you might want to breed your cats in the future, get them spayed and neutered anyway.
Yeah.
Just there are too many reproducing cats
Here, here.
running around.
Here, here.
And
And
animal
truth-
shelters, I know in Massachusetts this is true. I think this is probably true nationwide. They will not adopt out an animal without fixing it.
That's right. By the way, last night there was a reference on the debate to cats when Biden accused Trump of having the morals of an alley cat.
That is an insult to alley cats everywhere.
Yeah.
Exactly. That's what I thought. They're highly selective. Come on.
Maybe we could design a zinc finger AAV to turn off the reproductive
Yes.
gene.
Yeah. Well, people have tried coming up with various forms of birth control for wild and feral animals, and it's difficult. because they're really good at reproducing.
Yeah.
This is all true.
I mean, the AAV would have to be injected, so you'd have to
Yes.
capture them.
Yeah. And
All
if
right.
you capture them, you might as well just fix them.
Now you can do it by a dart gun.
Yeah.
I have an article from PBS News. Supreme Court allows cities to ban homeless people sleeping outside even when shelter space is lacking. Well, there it is, 6-3. Reversed
Seems
a
like a lot
ruling
of
by
decisions come down with those numbers.
yeah, not surprising. High Court reversed a ruling by a San Francisco-based appeals court that found outdoor sleeping bans amount to cruel and unusual punishment. I think this is unfortunate because if cities don't have shelters, where are the homeless going to go? And I think the Supreme Court needs to have a little bit of compassion for people. Not everyone is fortunate. Not everyone has a place to live or makes money, and you can have whatever opinion you have of them. Keep it to yourself because they don't have homes and they deserve to be treated properly. You know, in Salt Lake City, the local government does not like homeless people sleeping outside, so they give them places to live. That solves the problem. 650,000 homeless people in the U.S.
Yeah.
A lot.
Wow. Anyway, I find that very unfortunate, Supreme Court. We have 2 listener picks. Blog Design sends a YouTube video of our ally Paul Offit appears on PBS NewsHour to talk about how vaccine mandates are at play in the 2024 elections. And we mentioned this earlier. I think, Dixon, you mentioned it.
Yep.
And John writes, latest mammals infected with pathogenic avian influenza. This doesn't look good. So this is a USDA Animal and Plant Health Inspection Service article, detections of highly pathogenic avian influenza in mammals. It's a map showing different species in different states of the U.S. Look, the South doesn't have a lot yet.
They don't have a reporting system either.
We got cats, we have bears, we have dolphins, seals, coyotes, foxes, martens, mink.
Good Lord.
House mouse, raccoon, skunk, opossum.
Keep going.
Yeah, I'm sure this is just the beginning.
The
Yeah, it's
map
not
shows
great.
the list on the side.
Yep.
What's that?
The map shows.
He's not going to read the whole thing, I think. But yeah, there's a long, long list here of animals.
Yeah, it's a long list.
Species that have been found.
Just wanted to give you an example. It's 329 entries. Thank you for that. All right, that'll do it for TWiV 1127. You can find the show notes at microbe.tv/TWiV. You can send questions, comments, picks of the week to twiv@microbe.tv. And if you enjoy our discussions about science, please consider supporting us, microbe.tv/contribute. Dixon Despommier is at trichinella.org, thelivingriver.org. Thank you, Dixon.
Pleasure as always, as they would say. Definitely a pleasure.
Alan Dove is at turbidplaque.com, alandove.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.